Hello, I'd like to welcome and introduce Graceanne Tarsa. She's going to present her research with us today. Graceanne is a second-year master's student at the University of Wisconsin-Milwaukee, the School of Freshwater Sciences, where she works as a research assistant in Dr. Harvey Bootsma's lab. Graceanne received her Bachelor of Science in Biology and Math at St. Norbert College, and she began her work on Lake Michigan. During her time in the Bootsma lab, Graceanne has assisted with fieldwork as a certified diver on several projects and has also participated in outreach events that engage students and community members with the Great Lakes and the field of freshwater sciences. She hopes to combine both field research and outreach in her future career to work towards the conservation of aquatic ecosystems. Graceanne's current research is focused on the energetic role of round gobies in the nearshore zone of Lake Michigan. And today she'll be discussing quagga mussels, round gobies, and the nearshore food web. This past summer, Graceanne and Dr. Bootsma stayed in our dormitories in Sutton's Bay while they conducted their research in Good Harbor Bay. And we were really delighted to be able to offer them a space as researchers in the area to stay with us excuse me, in our new renovation, and we're really looking forward to hearing about her research today. So I will turn it over to Graceanne.
Well, thanks, Juliana, for the introduction and inviting me to present, and thank you to all the audience for being here today. Special thanks to the volunteers who are on today for all the work you guys do educating students and community members about Lake Michigan. I'm excited to share my work with you guys today. I actually grew up in Leelanau County on the southern end of Lake Leelanau, and living in Leelanau County, you are surrounded by water. And I spent a lot of time as a young student swimming and exploring the lakes and the rivers in the area. But it wasn't until I studied abroad in college that I really comprehended the impact that humans are having on aquatic systems. And when I got back, I had a new perspective for our freshwater resources. The Great Lakes have undergone a huge changes due to a variety of factors, including invasive species, human development, and climate change. And these will likely continue to cause change in the future. I love the water and want to work towards making sure everyone can love the water as much as I do. And I've been fortunate enough to end up at the University of Wisconsin Milwaukee School of Freshwater Sciences, where I'm able to study Lake Michigan. When I applied to work with Harvey, I didn't know that despite being located in Milwaukee, we actually do a lot of work on Good Harbor Bay right out of Leland with the support of the National Park Service. And that's been a really cool opportunity for me to learn so much more about the water I grew up in. And it's also a good gig because I usually have time to get ice cream with my grandparents when I'm in town. My work is focused on invasive round gobies and the energetic role that they have in the food web. But I have also been involved with some long-term monitoring and mussel removal projects during my time in the lab. And today I'm going to talk about our research regarding some of the changes that are occurring in the nearshore zone of Lake Michigan. I know that most of you guys have a pretty good understanding of some of the major players in Great Lakes health and the ecosystem, but I do want to start by highlighting some important background information just to get us all on the same page. Then we'll take a closer look at the impact of quagga mussels, the introduction of round gobies, and then I'll introduce my research project that I'm focusing on now and try to give you some preliminary results on that work. And we'll also then discuss some of the mussel removal projects that have taken place in Good Harbor Bay and finish with plenty of time for questions and discussion. The Great Lakes have gone through a series of really drastic changes over the last 2 centuries, largely due to both indirect and direct impacts of human development. As the communities and industries around the Great Lakes have grown, the lakes have been subject to pollution due to urban and agricultural runoff that's rich in fertilizers and pesticides, wastewater from sewage and water treatment plants, and point pollution from other factories. By 1960s, Lake Erie was kind of the poster child for polluted lakes. The pollution that was entering the lakes usually carries with it excess nutrients. In everyday language, we talk about nutrients in terms of nourishment, like proteins and vitamins, but when we're talking about nutrients in regards to the lake, we are really talking about chemical compounds that are supporting primary growth. And typically, we are most interested in nitrogen and phosphorus. In most aquatic systems, phosphorus is the limiting nutrient, meaning that it's the one that's most scarce and the one that plants and phytoplankton and algae are going to run out of first. So when we have excess nutrients that's entering the lake, it can cause massive algal blooms, and that's what we saw in Lake Erie and in other Great Lakes during this time. Often the algal blooms are toxic, and they also reduce the recreational value of the lake. But in addition to those, they also lead to hypoxic and anoxic conditions, which just means that the oxygen levels in the water are really low, and this creates dead zones that lead to fish kills. Colloquially, Lake Erie was sometimes referred to as the Dead Sea of North America, which is what this cartoon to the left is depicting. This degradation, though, didn't go unnoticed, and the other image we see here is community members protesting for cleaner water. Both of these images are from 1968 in the Cleveland Press, and by 1971, even Dr. Seuss was making note of the degraded system of Lake Erie, referring to it in his book, The Lorax, saying that the fish will walk on their fins in search of some water that isn't so smeary. I hear things are just as bad up in Lake Erie. In 1972, though, the Clean Water Act established pollution control programs, and the Great Lakes Water Quality Agreement established phosphorus loading regulations set to begin in 1976, and then it also defined individual lake targets. Over the next decade, phosphorus loading levels in Lake Erie and the other Great Lakes decreased, and we can see that in this figure here. Things did get less smeary, and Dr. Seuss, even with some encouragement, changed the text of his book to not include that Lake Erie reference. Unfortunately, before too long, and despite these still low phosphorus loading levels, the green decaying mats of algae began washing ashore in the 2000s. This bottom image is of the Manitou Islands, and this was a common on many of the beaches around Lake Michigan and the other Great Lakes. These mats consisted primarily of Cladophora, which is a form of nuisance benthic algae that grows kind of like hair on rocks and hard substrates, and it's common in many freshwater and even marine ecosystems. During the peak of growing season, July typically or August, nearshore rocky areas are often covered in this dense Cladophora growth, like this image we see to the right. But at this point, phosphorus loading levels are still low. They're beneath the targets, actually, and that prompts researchers to ask how nearshore systems are changing in a way that's promoting this excess benthic algae growth. Concurrent with the chemical pollution during the 1800s and 1900s, a form of biological pollution, if you will, was entering the Great Lakes through the introduction of invasive and more broadly non-indigenous species. Starting in the early and mid-1800s, the Wellington, Erie, and Illinois-Michigan canals were sequentially opened, connecting Lake Ontario and Erie to the Atlantic Ocean, and the Great Lakes chain the Mississippi River. The connection of the Great Lakes to the rest of the world was then finalized in 1959 with the opening of the St. Lawrence Seaway. What all of these channels had in common is that they allowed species that were not native to the Great Lakes Basin to enter either on their own power moving through these channels or through the ballast water of ships traveling from all parts of the world. It is important to note that not all the species that took up residence in the Great Lakes had large consequences, but many, such as the well-known alewife and sea lamprey, did, and during the mid-1900s, these species exploded in the region, devastating native species. One really notable one is the lake trout. The canals were also not the only way that species were entering the Great Lakes. Some were accidentally or even intentionally introduced. For example, in 1966, salmon were introduced in the Platte River in an attempt to actually control this booming alewife population. Soon after phosphorus regulations were put into place, policies and practices to minimize the impact of invasive species were also put into place. In the late 80s and early 90s, a lamprey-specific pesticide, actually called lampricide, successfully reduced the sea lamprey population, and official ballast water exchange and discharge regulations were also signed into effect at this time. Unfortunately, not all the species were as manageable as the sea lamprey, and new species introductions continued despite these regulations. Today, we actually have over 180 invasive and non-indigenous species in the Great Lakes Basin, and that prompts us to ask questions like, how are these invasive species impacting and changing the nearshore ecosystem? There are a few different ways to research an ecosystem and answer these kind of questions. One way is through long-term monitoring programs, and this is just what it sounds like. Long-term monitoring refers to data collection, data that's collected consistently at the same or a very similar location over an extended period of time. There are numerous of these sites across the Great Lakes, and they provide data that can be used to develop and test predictive models. They can be analyzed for trends, and they also prompt researchers to ask good questions about how things are changing. The Bootsma Lab has a long-time partnership with the National Park Service to perform long-term monitoring on Good Harbor Bay Reef. Over the years, the lab has actually done work throughout the park, including on the islands, but now we focus on a rocky site that's about 10 meters deep, and that is the GS10 site you're seeing in this map here. We also work extensively in the nearshore zone around Milwaukee, and similar there, we have worked at a variety of locations, but have settled on 2 rocky sites, one that's about 10 meters deep and one that's about 20 meters deep, and those are the ATW or Atwater sites. Each year at these sites, we deploy a buoy that's equipped with weather and water quality monitoring instruments. We put them out generally around May and retrieve generally around October, maybe a little later, depending on the weather. That data is actually accessible to everyone and provides real-time data on water quality and conditions, including wave height. So if you're headed out on the lake during the summer, you can go onto this website here that's listed on the bottom of the buoy image and check out the wave heights, the water temperature, and a variety of other parameters. We also deploy a number of underwater instruments that measure water currents, chemical parameters, and record some images. And some of those images are also available each day through the Great Lakes Observing System YouTube channel, this Gloss Buoy webcam site. We dive at each of these sites throughout the season to collect biological samples and service the equipment and instruments. And from these locations, we are also able to do more specific experimental research that complements this long-term monitoring data. All right, coming back to those questions we asked originally, in the late 1980s, Dracaena polymorpha, which is a species that's more commonly known as zebra mussels, were discovered in the Great Lakes region after presumably being introduced to the ballast water from transoceanic vessels traveling from the Pono-Caspian region. Zebra mussels quickly dominated the benthic, or the bottom region of the lake, in shallow nearshore areas, so typically areas that are less than like 20 to 30 meters deep. But the zebra mussels began to be replaced by a congeneric mussel commonly known as quagga mussels starting in the early 1990s. Quaggas are very similar, but they're a hardier and often larger mussel. Where zebra mussels prefer to attach to a hard substrate, quaggas are able to attach to soft substrates such as sand and silt when rocks and hard surfaces are not available. Quagga mussels are also more efficient in colder and deeper water, which has allowed them to expand their range in Lake Michigan and the other Great Lakes. By 2008, zebra mussel populations in Lake Michigan, which peaked in like 2002, 2004, were close to 0, and today quagga mussels blanket much of the lake bottom, as you can see in this video here. Zebra mussels are definitely not gone though. They are often the more prevalent species in warmer bodies of water, and we actually find more zebra mussels than quagga mussels in the harbor slip right off of Lake Michigan, where the university docks our research vessels. Quagga mussels play a really unique role in the food web as efficient filter feeders, which means they feed by drawing in the surrounding water and extracting the phytoplankton, the particulate matter, and other organic compounds. The image to the left is an image of a mussel with both the in-current and ex-current siphons extended and open. And if you're observing them, you can actually see water entering through that in-current siphon. When startled, the mussels will pull those in and close up for protection, which is what we see here when the diver is approaching and then touching these mussels. Just like other filters, the mussels will strip the water of material and in the process make the water more clear. And that's what this image on the left is depicting. Both of these chambers were filled with harbor water. The right chamber has mussels where the left chamber does not. And after just 30 minutes, you can see a drastic difference in water clarity due to this filter feeding behavior. The no mussel chamber does have some change in clarity due to the settling of the particulate matter, but there's still a large difference then between the no mussel control and the mussel chamber. On a lake-wide scale, an increase in water clarity was documented just 10 to 15 years after the initial invasion of Dracaenids, and this is another good example of the importance of long-term monitoring. Secchi disk depth can vary day to day and even year to year, but Secchi disk measurements that are taken over an extended period indicate these trends and patterns. And in this case, we see a pretty substantial increase in Secchi disk depth, which is translating to an increase in water clarity. When mussels are filter feeding, some of the harvested energy is invested as new mussel biomass, but a large portion is actually egested as feces or pseudofeces, which are collectively referred to as biodeposits. This is problematic because that egested nutrient-rich material concentrates in the benthic region, again, the bottom region of the lake, where it can be readily used by detritivores and benthic algae, and ultimately, this cycle redistributes nutrients and likely contaminants from the pelagic, the open water area, to the benthic communities. We talked about the importance of phosphorus a bit earlier, and if we pretend these little red dots are each phosphorus that is bioavailable or available for plants to use, we see that we have some phosphorus entering through this top and some that's exiting in this outlet. And we have some phosphorus that is settling out of the water column. And those are those larger red dots. But most of the phosphorus is staying in this pelagic zone, in this area that's open water, that's accessible to organisms. When the quagga mussels enter, they begin pulling down the water and filtering it out. And we see that more of the phosphorus is being shifted towards the benthic region where it's more accessible for the detritivores and benthic algae. And after a while, the quagga mussels have actually concentrated all of that phosphorus in that bottom layer, and now it's not in this open water habitat where it would be accessible to phytoplankton. And this redistribution phenomenon for phosphorus has been termed the phosphorus shunt. So now we have quagga mussels that are increasing water clarity due to filter feeding, and in doing so, they're actually increasing the depth at which photosynthesis can occur, and thus increasing the possible habitat range for benthic algae. And even in the areas where there was already enough light for photosynthesis, mussel beds are actually creating a rough substrate that's increasing the available surface area for the benthic algae growth. On top of this, we have this redistribution of phosphorus into the benthic region, making it accessible for benthic algae. And while the mussels are down there doing their thing, they're also respiring, which is producing a readily available CO2 source, which is required for benthic algae photosynthesis. So under these conditions, it's not surprising that we see this resurgence of cladophora production. This figure here depicts cladophora biomass over a summer season when mussels are present and when they are not. And you can see that the cladophora biomass when mussels are present, which is the solid line, and that's at a density that's pretty common on Good Harbor Bay Reef. It's about 3 times that of the cladophora biomass when no mussels are present in that dashed line. To put these values in a little bit in perspective, the tentative target that we're aiming for is about 50 grams of dry weight per meter squared. Like all things, you can identify pros and cons of the mussel invasion. A pro, and the one we often most hear, is built on the idea that a clear lake is a good lake. It's clean, it's nice Caribbean blue in the summertime, visibility underwater is higher, and while this all sounds really great, we have cons like this increased cladophora growth and reduced phytoplankton and other important zooplankton species, which are important for supporting fish populations, and as a result, the fishing industry, which the Great Lakes communities rely on. So just like the questions we ask regarding the phosphorus cycling, you can ask the same thing about energy cycling. We have seen that mussels have created this phosphorus shunt. What about an energy shunt? Are mussels a trophic dead end, or is that energy being passed up the food web? And if So if they're not a trophic dead end, how is energy being recycled and passed up the food web to higher trophic levels, including birds and fish? Unfortunately, quagga mussels are not an easy food source to take advantage of, and despite being abundant, they're actually generally unused by native species. First, if you've ever encountered quagga mussels, you know they have a really hard shell, and to harvest them as a food source, a species must possess specialized pharyngeal teeth to crush that shell, and that's an adaptation that's typical of efficient molluscivores, organisms that eat mollusks or mussels, but it's uncommon in other fish species. Quagga mussels also have byssal threads, which are strong, sticky threads that they use to attach to surfaces, and that's what we're seeing in this top left image. As the mussels grow, so does the number of byssal threads, making them very difficult to remove from the rocks. And when we collect these when we're diving, we actually use paint scrapers to remove them, and even that can be tough, and you can see the divers in this video struggling to remove them from those rocks. Lastly, quagga mussels grow in these tight clumps. They will even attach to each other or the dead shells of other mussels, and that creates lots of tiny crevices for small mussels to shelter in, and it also just makes the mussels harder to grab on individually. In 1990, right after the introduction of the Graceanne mussels, another invasive Eastern European species, round gobies, was discovered in the Great Lakes Basin. Specifically, it was discovered in the St. Clair River. If you are unfamiliar with round gobies, they're a small benthic fish, again, bottom fish, that typically is found in the rocky or gravel substrate of shallow areas. And as Graceanne mussels and round gobies are native to the same area geographically, gobies actually have several adaptive advantages that enable them to predate upon these Graceanne mussels. Round gobies do possess that specialized pharyngeal jaw that allows them to crush the mussels. They also possess no swim bladder, which typically allows fish to control their buoyancy. And as a result, gobies are demersal, so that means they live on the bottom their entire lives, providing them great access to these benthic mussels. Despite gobies' inefficiency at harvesting clump mussels or those with abundant byssal threads like quaggas, they've actually been found to preferentially choose Graceanne mussels over native mussels and clams. And this adaptation to access Graceannes as an energy source is likely one reason they have been so prolific since their invasion. With that said, there are a lot of other factors too. Round gobies are versatile and can adapt to varying environmental conditions, and within 10 years of their discovery in the Great Lakes, populations of round gobies could be found in river mouths, harbors, and even open water habitats. Gobies also survive in low water quality, including low oxygen levels, and even in brackish water systems. They're also fairly aggressive. They have long spawning season and lack natural parasites in the Great Lakes. And this combination has allowed round gobies to quickly establish themselves in all 5 Laurentian Great Lakes, with especially high concentrations in Lake Erie and southern Lake Michigan. As their populations have grown, round gobies have outcompeted some native species, most notably the molted sculpin, and have forced others to change their behavior. So now, 30 years post-invasion, they are noted as the most prominent prey fish in some nearshore areas. The abundance of round gobies and their consumption of Graceanne mussels has made round gobies a critical aspect of management plans. As we talked about prior to the goby invasion, few organisms fed on these Graceanne mussels, and as a result, the energy, nutrients, and contaminants that was sequestered by mussels was essentially lost to the ecosystem. Post-invasion, however, gobies have created this channel for those energy, nutrients, and contaminants to pass to higher trophic levels. In the Great Lakes region, round goby predators include many fish species, such as yellow perch, lake trout, and walleye, and they have also a common food source for an assemblage of bird species, some of which are known to die of avian botulism, and these include cormorants, gulls, and loons, and mergansers, as we see in this picture here. Some predators, such as the lake trout and the brown trout, have been found to be increasing their reliance on gobies in the recent years. And since gobies are this abundant and generally easy to capture prey source, predators that rely on gobies as a food source have been found to experience high growth rates. One example of this is the Lake Erie water snake, which has recovered from near extinction by shifting to a diet that's more than 92% composed of round gobies. Personally, I'm not thrilled about this, but all of God's creatures have a place in the choir, right? Before we go on, I want to take a second to review all these connections and kind of create one storyline. This is going to be just the bare bones, but let's start with the nutrients entering the water. And it's important to point out that not all nutrients are bad. Lakes need nutrients. Nutrients are natural. We run into problems when we have excess nutrients. So we have this nitrogen and phosphorus entering the water column, and that can be used by phytoplankton to grow. Phytoplankton are then consumed by zooplankton, who are consumed by small fish and birds and larger fish. And this energy is passed up the trophic level in a fairly well understood pattern. Now enter the quagga mussels, which are going to consume a large portion of the phytoplankton. The zooplankton pathways are still present. These have not completely disappeared, but it's important to note that this zooplankton pathway has been greatly reduced. As we talked about, these quagga mussels are going to promote the growth of cladophora. The cladophora is not going to be directly consuming the mussels, so this energy is still staying with the quagga mussels. And you can see there are no large arrows indicating that this energy is passing up the food web, and we have this trophic dead end almost. Round gobies are then changing that. They can eat the quagga mussels, and then other organisms up the trophic food chain are able to consume round gobies. But there's still a lot of questions surrounding this connection here between the benthic nearshore and these upper trophic levels, and how round gobies are impacting that. Because round gobies are this prominent prey species, a better understanding of that energetic role in the food web is needed, and that's the project I'm currently working on. The broad objective of my research is to quantify round goby productivity in the rocky nearshore zone of Lake Michigan. What I mean by this is I want to put a number to the rate of production of new biomass and the energy associated with it that can then be theoretically passed on to higher trophic levels. Productivity estimates depend on several population and bioenergetic parameters. Population estimates are pretty straightforward and can be made by combining individual measurements of length and mass from gobies that we collect at each of these sites with the size distribution and density measurements from video transects that we collect while we're diving. These measurements over time can tell us how the round goby population is changing, which could inform how other populations that rely on round gobies are managed. Long-term data has been collected on Good Harbor Bay for goby density, and from that data, we see an overall negative trend, at least through 2019. We haven't finished processing the 2020 data yet. On a monthly basis throughout the season, we see a positive trend in density, with July through October remaining fairly consistent. There is quite a bit of variability within the size classes, but we do see an increase in small gobies over this time. It could be that they're spawning early in the season and now they're big enough for us to see, or just big enough to be hiding less and be out looking for food more. The population parameters also allow us to compare goby populations spatially between sites, temporally through the season, or even compare subgroups within a population, such as males and females. One way to compare this is through length weight regressions, and these are used for a lot of species of fish. The graph here on the left is just a raw compilation of gobies that we collected this year. These data can be fit with an exponential regression that can be used to estimate the weight of gobies when we only know their length, such as from video transects. So by combining data from these regressions with those video transects, we should be able to estimate total biomass. The length and weight data can also be used to tell us about the condition of a fish. Because mass is exponentially related to length, we use a condition factor K, and that's the equation we see at the top of the page. A higher K value is a fish that's fatter or bulkier, more likely healthier. Again, this is very preliminary data, but throughout the season, we have observed very little change in the condition factor of gobies on Good Harbor Bay Reef. While the biomass measurements are important, we have to distinguish between biomass and productivity. So instead of talking about round gobies for a second, we're going to switch to apple trees. Both of these apple trees have the same biomass. The difference is that the apple tree on the left is only producing 1 new apple a year, while the apple tree on the right produces 3 new apples a year. So when Hungry Rudolph is looking for a good source of food, he's going to want to rely on this right tree. He's going to want one that's going to have a high production to biomass ratio. And the same is going to go for large fish and birds who are looking for good food sources in the water. So the biomass measurements are only telling really part of the story, but with that said, they are a really important part of the story because the abundance and density measurements can be combined with individual measurements of growth and feeding rate to estimate those parameters on a population level, and those are the productivity estimate measurements that we are going to be more interested in. Where the abundance and density measurements are pretty straightforward, some of the bioenergetic variables, such as growth and feeding rate, are more complicated to measure and also a little bit more variable. And we're going to start by talking about growth. Close examination of scales and other bony structures, such as spines and vertebrae, allow for the age and growth history of fish to be determined. I originally tried working with scales, but they're really difficult to interpret, so I ended up switching to otoliths, which are small ear bones that help fish orient themselves and maintain balance. And an example of those is shown in these images here. When an otolith is first developed, that becomes the center of the growing fish's otolith, and that's called the focus. As the fish grows, concentric circular rings or ridges called circuli develop around the focus on the edge of the otolith. And in this image, each thicker band that we see consists of many thinner circuli. During periods of rapid growth, such as during ideal summer conditions, when feeding rates are high, the circuli are spaced apart and create this translucent region. These otoliths are mounted on dark background, so the rapid growth region is actually going to be these darker rings. When growth slows, like during the winter season, the circuli are going to grow very tightly together, and that creates a region called an annulus. And this is the opaque region we see in these images. Since true annuli only form once a year, the age of a fish can be determined by counting the number of annual rings present, very similar to how you would count the rings on a tree if you were trying to age it. In these examples, we can start at the center and count our way out, and we are looking for the area where a dark region switches to a light region. So starting on this left one, we choose our first 1, 2, and then 3, and we would determine that this is a fish that's 3 years old. The one on the right is a little more complicated because it has a lot more rings, but we can follow a similar pattern, counting these concentric circles, 4, 5, and we would make the determination that this goby is 5 years old. This is very, very preliminary data and does not include very many replicates. It's also not fit with a growth model, but eventually I'm hoping that the data on the left will look more like the data on the right. You can see that there is quite a bit of variation in length at each age, and I expect that once we have more replicates, we may have a few outliers that are either extra large or extra small for their age for some reason, and we also might find that there are some otoliths that were misinterpreted, and then we'll be able to remove those from our analysis. We do expect that some of this variability will remain. Just like humans, fish are going to grow at different rates. Eventually, we will fit this data with a growth curve, and then we'll be able to determine several variables about the growth patterns. The asymptotic growth, which will be the theoretical max size that the average goby will approach. And then we'll also be able to determine growth rate at different ages. Based on this very preliminary data, we can see that it looks like growth is most rapid during the first year, which is consistent with past research, and we expect that pattern to be the same once we have added more replicates to this. Once we know how many gobies there are and how fast they are growing, we want to put that into an ecosystem perspective because that's really what's going to be informative for managers. Round gobies serve as this energetic connection from the benthic nearshore to the upper trophic levels, and we can kind of fit them in with either the higher trophic levels or the lower trophic levels separately. As we saw, there are many known predators of round gobies, and studies that are regarding the proportion of a predator's diet that is composed of round gobies. And those studies help to fit round gobies in with the upper trophic levels in the food web. Fitting them in with lower trophic levels is also important. It can be broken down into 2 smaller questions. How much are gobies eating, and what are gobies eating? Accurate feeding rates can be coupled with benthic invertebrate and Dracaenan mussel abundance measurements, as well as with diet composition data to estimate environmental carrying capacity and the flow of energy from lower trophic levels to round gobies. Unfortunately, how much round gobies are eating is a really tough question to answer. There are some estimates for mussels, but as we're going to talk about in a minute, gobies are consuming soft-bodied invertebrates, and it's possible that this consumption rate is different for those soft-bodied versus the mussels. What I'm trying to do is combine measurements of how much gobies have in their gut when they're captured, and then compare that to measures of gastric evacuation rate from lab experiments. So I've set up a series of lab experiments to measure how long that food is staying in the gut, and the right picture is one of those experimental setups. The left picture is some round gobies that we collected and I housed in the lab for those feeding experiments. This work is a work in progress, and right now I don't have any results. Hopefully we'll get there. What gobies are eating is also important for determining an environment's carrying capacity, and it can tell us about how energy is moving, which could be important when considering the impact of round gobies on mussel populations, as well as the impact of mussel removal projects on goby populations. It's nice too, because what gobies are eating is a little bit more straightforward to answer. One way to do it is to analyze the gut content, which can be misleading in some cases because food types such as mussel shells are not only more recognizable in the gut, but they also likely have a longer residence time, so their importance in the diet can be overestimated. Another way to answer this question is to look at gobies' stable isotopes, specifically carbon and nitrogen isotopes, as these follow really predictable patterns in the food web. Carbon can tell us about the primary energy source gobies rely on. In the nearshore zone, this will either be dracaenid grazing of phytoplankton or soft-bodied invertebrates that are feeding on benthic algae. The nitrogen isotopes can also tell us about their trophic position, as well as the trophic position of the cladophora, the mussels, and the benthic invertebrates, because we'll analyze that as well. The consumption of mussels as any part of the round goby diet does naturally raise questions regarding the viability of gobies as a management tool for mussel populations. Early research has concluded that gobies are unlikely to completely remove mussels from a given habitat due to several inefficiencies. First, gobies lack that swim bladder, which does provide them the advantage of feeding on the mussels, but it also means that mussels that are vertically segregated in the water column, such as those atop extra large rocks or on underwater structures, are going to be inaccessible. Research has found that though gobies try to feed on larger mussels, there's a significant size difference between mussels removed by gobies and those that are attempted to be but not successfully removed. Those larger mussels tend to have an increased abundance of strong byssal threads and a thicker shell, which could explain why gobies are preferentially consuming mussels that are typically 13 millimeters in length or smaller. Gobies are also inefficient at removing and consuming mussels located in tight crevices within large clumps or hidden on the underside of rocks. So small mussels can avoid predation by taking refuge in these locations and then migrate once they're big enough to avoid predation on their own. Even if complete mussel removal by gobies is unlikely, round gobies still may have a potential ability to impact mussel abundance. Several studies have supported this. In Green Bay Lake, Michigan, which is the figure we're seeing on the right, studies have found dramatically fewer mussels in areas where gobies have become established and a statistically significant negative relationship between round goby density and mussel abundance. In eastern Lake Erie, round gobies were also found to reduce dracaenid populations by nearly 94% from 2001 to 2004. But unfortunately, while they may cause declines in localized mussel populations, studies have found that consumption rates are likely not high enough to affect dracaenid populations on a system-wide scale. Even if they can't necessarily cause these decline in mussels, maybe gobies could control mussel populations or keep certain areas mussel-free if mussels could be removed by other means. In 2016, a 40-meter squared plot of lake bottom at Good Harbor Bay Reef in Lake Michigan was manually scraped by divers to remove the mussels. And since then, we have monitored the site and no new mussel populations have become established in this relatively small plot, which has led to some interesting questions regarding goby predation, which is what we hypothesize is keeping mussels from becoming recolonized in this area. To test that hypothesis, goby exclusion experiments were deployed in May of 2019 and then were monitored over the next few months and into 2020. Some of those fell over due to strong windstorms, and others were covered in cladastra, so our results weren't conclusive. But we are currently exploring this question a little bit further with Hester dendy samplers in the lake. That's this image we're seeing to the right, and these create really small crevices that are too small for the gobies to get into. We're still monitoring these, and we'll monitor them in 2021, but by the end of the 2020 field season, there were several mussels observed within the sampler, indicating that yes, gobies could be responsible for keeping the scraped area clear of new mussel growth. In 2019, Zequinox, which is a dracaenid-specific molluscicide, was applied to a 300-meter squared plot of lake bottom near the 2016 removal site. After a month, mussel biomass had decreased by nearly 90%, but by a year later, this area had had some recolonization of dracaenid mussels. In this figure on the bottom left, each bar represents mussel density, or the number of mussels per meter squared on each sampling day. The blue bars are the control site where no Zequinox was applied, and the orange bars are the treatment site. August 2nd, which is this first set of bars, was pre-treatment, and the mussel density between the control and the treatment was pretty similar. The next set of bars are 2, 6, and 30 days post-treatment, and the control site, again, which is the blue bars, remains consistent throughout this period, but the mussel density on the treatment site is significantly reduced. In 2020, the Zequinox site still has lower mussel density than the control site, but we do see some increase, which is suggesting that we do have some recolonization. This is a bit surprising because that 2016 site has stayed clear over several years, but the thinking is that the chemical application may not have effectively killed mussels on the sides of rocks, which is an area that's less sampled. Typically we sample the top of rocks, and you can see the application method in this top image. We put down structures that are about 1 meter high and covered them in tarp, and then injected the Zequinox. So as that Zequinox is settling, it's likely settling on the tops of rocks and not sticking on the sides of rocks. The thinking is that because those sides of rocks are surviving, those mussels were able to migrate to the top and recolonize those areas. In some cases, those mussels were probably too large for gobies to consume. When those mussels reproduced, they're able to then provide shelter for the smaller mussels until they're able to avoid predation themselves. We're only a year out from this project, and we'll continue to monitor it. And in the meantime, we're also trying to brainstorm other ways that are maybe less expensive or less labor intensive, but still successfully remove mussels, hopefully from the tops and the sides of rocks. What all of this is to say is that the Great Lakes are a continually changing ecosystem due to a variety of factors, including invasive species, human development, and climate change. In the nearshore zone specifically, invasive quagga mussels and round gobies have had a significant impact on nutrient cycling and the food web. We are going to continue to rely, if we are going to continue to rely on Lake Michigan and the Great Lakes for resources, recreation, and commerce, we need to understand the changes that are occurring and how the ecosystem is responding. And one way to do that is through research, including both long-term monitoring like the buoy in Good Harbor Bay, and by asking and answering specific questions such as the impact of round gobies and quagga mussels. The data we collect and the conclusions we draw can then be shared with management agencies, regulators, and communities to develop models and management strategies that influence policy decisions and hopefully eventually then change behaviors. This work takes a lot of collaborators' brainpower and time. First and foremost, my advisor, Harvey Bootsma, whose years of persistent and novel work have led to some really important findings and publications. My lab mates who help with sample processing and fieldwork. The National Park Service has been really instrumental in funding this work and helping with fieldwork in Sleeping Bear. The Michigan Department of Natural Resources has also does a lot of really complementary work in the area, and they help extensively with the fieldwork as well as with some sample processing. There's also a really great group of volunteer and contracted divers who help us with the biological sampling, especially when we're unable to make trips over from Wisconsin. And last but certainly not least, Inland Seas has also been an important collaborator through their work in the area and have been generous to share the Thomas Kelly Biological Station lab space with us during our trips. With that, I'd like to thank you guys again for joining today and open it up for any questions or discussion points that you guys may have.
Wonderful. Well, thank you. That was great information. I think science communication is so important, and you had so many great visuals there to take these really complex ideas about freshwater ecosystems and really make them accessible. So thank you for sharing with us. So yeah, we have some time for questions now. We have a question from Susan Odgers, and she is asking, how will you be working, if you will be working, with the Traverse City Fish Pass? So do you have any connection with the Fish Pass project in Traverse City?
I'm not familiar with the Traverse City Fish Pass project.
It's not related. If you have any questions, you can either put them in the chat, or you're welcome to unmute yourself if that's easier to ask a question.
I'd like to ask a question. Graceanne, what did you use to collect the gobies that you used for your experiments?
We used a variety of methods, primarily gill nets, which we deployed while we were diving, and then we were actually able to remove the gobies from the gill nets underwater to make sure we were keeping them alive and in good condition to bring back to the lab for the experiments.
Yeah, I was wondering because from the pictures, those are pretty big gobies, and we don't usually see those in our trawls.
Yeah, they are pretty big gobies. We did collect a few really small gobies using hand nets, but most of them were a little bit larger because they were the ones that were getting stuck in the gill net.
Do you have a sense of the average size?
Yeah, we're seeing a lot that are like the 7 to 12 centimeters long.
Okay.
It really depends on when we're out sampling. We were out in August near Milwaukee, and we almost saw no big gobies. We saw ones that were 6 centimeters or less almost the entire time. So it's going to depend on when you're out, but generally we're seeing about 7 to 12.
Great.
Thanks for your presentation. How large is the scraping site?
The 2016 manual removal site was about 40 square meters. The Zequinox site was about 300 square meters. It was a 30 by 10 meter plot. So that plot, if you're in the center of it, you're never more than 5 meters away from one of the edges.
Thank you.
I have a question. You mentioned the quagga mussels can migrate, and I was curious about how they go about doing that. I always assumed that that mussels were pretty much stuck to wherever they landed. So how do they do that once they've already put down byssal threads?
Yeah, I'm not sure if quagga mussels are using byssal threads or their mussel foot. Most mussels have a pseudo foot that they're actually using to move, but they're able to detach those byssal threads and move. The quagga mussels are actually moving. If you look at videos or time-lapse images over a series of a couple hours, the mussels are actually moving a pretty substantial amount, more than you would think just looking at them.
Okay, thank you.
All right, we have a question from Mike Foley. He asks, in an early slide, you showed a decrease in goby density over the last few years. Is that thought to be related to increased predation by other fish species?
It's hard to tell based on the data we have. So there are a couple ideas. It could be due to predation by other fish species. It could be due to just round gobies kind of overshooting their limit in the environment and then kind of coming back down to a more steady state. It also could be that they're controlled by how much food they have available to them. So I'm not able to say exactly what's causing that decline, but there are kind of several possibilities that we could explore further with research.
And Jillian asks, in the feeding experiments, you had a video with a goby eating something that sank down in front of it. What was that, and what do you feed them for the feeding experiment?
Yeah, that's been part of the problem, is trying to find a good food source for them. In the video, I was feeding them mussel tissue. So I took mussels, dead quagga mussels, opened them up, removed that soft body from inside, and then I actually dyed it. The idea of dyeing it is that it'll be easier to see when it's in their gut and also when they've passed it through their gut as I'm looking at gastric evacuation rate. I've also experimented with feeding them chironomids. The problem with that is the chironomids I was using are freeze-dried and they float on the top. And since gobies are benthic fish, I really need the food to sink. So in addition to the quagga mussel soft body tissue, when I'm just having the gobies in the lab, keeping them for a couple weeks before I start these feeding experiments, I'm using just a pelletized fish food that's based on insect larval based pelletized fish food, and it's a sinking food, so it will slowly sink from the surface all the way down to the bottom, and then they'll eat it. That goby was really excited to eat. They're not all that excited to eat, but sometimes they're pretty excited for breakfast in the morning.
I'd like to ask a question.
Go ahead, Don.
And I know your work is specifically tied to the Lake Michigan nearshore systems, but over the years up here in some of the inland lakes, we certainly noticed a large influx of the zebra mussels, and they have, to my knowledge and my contacts, they've continued, but to my knowledge, none of the inland lakes that it was quite a few up here where I was contacting people, did they end up seeing quagga mussels? And I don't get it. They were brought in, we're pretty sure they had to be brought in by boats and, their bilge water and so on, and the trailers.
Yep.
And so I'd wondered if you knew anything about that at all. And if you don't, I understand.
No, you're right. Most of the inland lakes that have zebra mussels, it's because of them being brought in on some kind of boat, in water that's trapped in the boat, attached to the hull, attached to the trailer, something like that. The zebra mussels are equally as good, if not better, in that warmer water or the faster moving water, which is probably why we haven't seen the quagga mussels dominate the area as much. It's a little bit different in Lake Michigan because it is so much deeper and colder. And in those instances, the quagga mussels have this advantage that allows them to be better than the zebra mussels. So my guess, and I'm not positive on this, would be that quagga mussels have probably been introduced accidentally to these lakes, but because the zebra mussels were already established and they don't have any disadvantages in that warmer water compared to quagga mussels, they're doing just fine in the inland lakes.
Okay.
We had a question about human behavior. So what are things that the public can do in terms of this issue?
Yeah. So this is interesting because it's not like any of us are going to go out there and catch all the round gobies. We're not going to go out there and remove every mussel. So the biggest things we can do is making sure we're not transporting them to a new inland lake. There are a lot of lakes that don't have zebra mussels in them. There are a lot of lakes that don't have round gobies in them. So keeping those lakes in their natural state is our responsibility. And the best way to do that is through monitoring and education and making sure people know what aquatic invasive species are, the impacts that aquatic invasive species have, and how they can minimize the transport of aquatic invasive species. So by cleaning off boat trailers, by making sure we're not releasing fish that we catch in Lake Michigan into inland lakes, by making sure we're not transferring via bait buckets. Those are all things that we can do on a personal level to make sure we're not making the problem worse in the inland lakes.
All right, we had another question. Does the Zequinox affect the cladophora? And if not, does the cladophora hide resurgent quagga mussels?
So in our sampling, we will remove the cladophora as well as the quagga mussels to make sure that if the cladophora is hiding any, we do find those quagga mussels. As we kind of saw in one of those figures, mussels are helping this resurgent of cladophora. So when you have a mussel-free area, we would think that there would be less cladophora because they don't have access to all of that extra nutrients. We are monitoring that at our 2016 scrape site. The Zequinox, during the application period, we did see a reduction in both soft-bodied benthic invertebrates and Cladophora. It's possible that this is just due to the destruction of the habitat. So because the quagga mussels were dying and falling off the rocks, all of the Cladophora that was then attached to those mussels is also going to fall off. And kind of the same thing with the soft-bodied benthic invertebrates, that any of those that were making their homes in the quagga mussels colonies are also going to then fall off the rocks and not survive during the Zequinox application. We have seen a resurgence in Cladophora growth at that Zequinox site, though.
Yeah, go ahead.
Ask a question. Graceanne, at the very beginning, you talked about phosphorus and nitrogen. So is there anything that we as homeowners near the lakes can do? I'm thinking in terms of landscape, caring for lawns and that kind of thing.
That's a really good question. One of the things, and this is a big hot thing in Milwaukee right now, is dealing with rain barrels. So when water is coming, when it's raining and we're seeing, or it's snow melt, having some collection method so that all of that water is not running directly from your driveway to the street and down to the lake, that it's going through the earth in some kind of natural filtration system. It's also important that we can minimize the amount of fertilizer we're putting on our lawns, the amount of salt we're putting on driveways, all of those kind of things. Obviously, not everyone is able to make that zero, and that's okay. If we're moderating that and making it smaller, that's something that even individual homeowners can do as well.
Great question. Jillian is wondering, what do the gobies do in the winter months? Is anybody studying them during the winter?
It's really hard to study them in the winter because no one wants to dive in the winter, and I completely understand because it gets pretty darn chilly down there. The gobies are actually going to migrate to deeper water typically in the winter time. So where we see them at like a 10-meter site, they're going to go down to like 20, 40 meters even, and just kind of hunker down. When we have upwelling events, which are times when the water is shifting in such a way that all this cool water is coming up near shore, and that happens a lot on the Milwaukee side, the gobies in those colder conditions change their behavior. So when we're diving, we may not see very many gobies, and when we do, they're very skittish, they hide a lot. So that's kind of their behavior in the winter time as well when they're at those deeper sites.
And we have another question here. The Cladophora fueling emissions from mussels are solubles. What solubles do you place higher on the influence hierarchy for algae growth?
I'm not sure if I fully understand that question.
Len, would you like to clarify your question at all?
Yeah, Harvey and I have exchanged a lot of correspondence on soluble reactive phosphorus. Mm-hmm. And then in other studies, ammonia has been highlighted as a significant fuel. And you just mentioned that the emission of carbon dioxide is also a factor in encouraging Cladophora growth. Is that the whole influence environment, if you will, to encourage Cladophora or other algal growth?
The main nutrient that's going to be promoting that algal growth is most likely phosphorus, that soluble reactive phosphorus, because phosphorus is the limiting nutrient in most aquatic systems. Mm-hmm. So nitrogen can be fixed by some organisms, they can get it from the atmosphere, but we're not going to be getting phosphorus from the atmosphere or other sources. So when the Cladophora, when the mussels are ingesting this phosphorus-rich material, that's really what's going to be helping the Cladophora growth. There are other factors like the water clarity and the CO2, but the phosphorus is going to be the big one.
I've got a question. So at Inland Seas, we're always trying to inspire the next generation of freshwater stewards. And I'm curious what inspired you to go into freshwater sciences?
What inspired me and what I always tell students when I'm talking to them about what I do as a job is that I just had the opportunity as a child to spend a lot of time in the water. My grandparents had a pool. My dad always was taking us to the lake, and typically not like the beaches. He would just take us to a lake and we would go swimming. So I spent a lot of time just in the water, exploring it, finding things and being curious about them. And that's what really made me interested in it. Living in Leelanau County and in the Grand Traverse area, there are a lot of opportunities, even for like young students, to get involved with river monitoring on like the Boardman River or the Grass River and stuff like that. And so I got involved with those. And that's where where my interest in like a more scientific approach came from. But generally just the interest in water was just from spending so much time in the water.
Wonderful, thank you. Do we have any last questions?
I've got one more, Graceanne.
Mhm.
Did you notice in your tests if there was actually a size limit of the quagga mussels that the gobies could eat?
So past research I think is like 13 millimeters is what most research is saying. I'm not sure. We were finding some very large gobies, 15, 16 centimeters.
Mhm.
And my guess is that they are actually able to consume mussels that are a little bit larger. They may preferentially choose those smaller mussels even still. I'm not sure if it's known what the exact size limit of mussels that the gobies are able to consume is.
Thank you so much for your time and sharing all your research with us, Graceanne. We're excited to see where it goes and what you learn. And thanks to everyone who was on here today. And I hope you all have a good new year.